presentation.
Can you hear me well?
Yes, we can hear you.
Okay, perfect. So hello, everyone. I'm really happy to be a part of the program. And thank you, Dr. Stephanie, for the introduction. So my name is Annie Elong. I'm a research instructor at La Jolla Institute for Immunology. And I'm working mostly on emerging viruses. And today I just want to share with you first my education. So what led me to be where I am here now, and also talk a bit about the research I'm doing. And I think it will be a nice introduction to virology and immunology and how we can combine both of those disciplines. So let's go. And of course, I want it to be interactive. So if you have any questions, just feel free to unmute yourself or put on the chat, and we will have a nice discussion, I'm sure. So just a little bit about me. I'm coming from France, so I did a lot of my study in a city called Nantes, which is in the northwest of France. And I decided to come in a night to stay in San Diego, the beautiful San Diego. I really like the sunset, so that's why I put a picture of the sunset, and particularly at La Jolla Institute for Immunology, to work on the development of cutting-edge approaches to study and tackle infectious disease worldwide. To talk about how I became a scientist in space and time, it started when I was little in a country in Central Africa called Cameroon. I had, when I was around 8, 10 years old, I was dreaming about being a medical doctor. And I don't know for you, but it was the thing. If you ask me, what do you want to be later? I will say medical doctor for sure. And I will tell you why after. And in the time and space, I moved to France, where I did a big part of my education, like I say. And during my high school, I started to be interested by biology. And I did a bachelor in biomedical science. And after that, a PhD. I fell in love with immunology. So I did a PhD in immunology. And of course, as you know already, I moved to US to work on virology, immunology, and now I'm a research instructor. Just for your information, research instructor, it's after a postdoctoral fellowship. It's a transition to be a principal investigator, so someone who will have his lab and do his own research without having a supervisor. For the moment, as a research instructor, I still in the lab of someone else, and this person is my supervisor and supervise my project, even if I'm supervising other people. So we can discuss more about that. And also one thing I put on the presentation, it's from my senior high school to my PhD, I did 8 years. But you will see that it's really fast, because even when I was doing my bachelor, I didn't know I will do a PhD, but when people were talking about doing a PhD, I was like, no, that's too long for me. But actually it's not too long. And why? Why did I choose biology? And this is one of the pictures I like. Actually, it was in our living room. And I wanted to be medical doctor because I really wanted to be a particular type of doctor. So the one without border, because I was seeing at the TV all of the disease around us, and also the natural disaster. And what I was thinking is it will be great for me to help people and fight the fear of disease and also the fear of natural disasters. And we know that after natural disasters in general, there is also a bunch of medical and health issues that are going on. But during my courses, I met a lot of people from different backgrounds, and I had the bright idea to check biomedical research. And this is where I am now. And I understood that with biomedical research, I was able to understand disease, develop vaccine and drugs, so helping also people and fighting the fear of disease and disaster, as natural disaster can bring also health issues. And one thing that I want to share with you today, and I think it's really amazing for to be in high school for the majority of you and to be able to have this kind of boot camp, because I didn't have it. And I think I missed a lot of opportunities to really know what I wanted to do exactly or what were my options. And I think now the type of advice I can give is to be curious. It's really important to be amazed by everything around you and ask questions. It's important also to talk to people around you, to your parents, the parents of your friends, your relatives, the extended family, people that you don't know but you are curious about. Just talk to people. I know it's not easy, but it's something really helpful and it will help you for your career as well. And very early in your education, you should look for your interests. When I was in high school, I was interested by biology. I knew it. I knew I want to understand the complexity of the human body, but I was not good doing biology. I was good doing physics, chemistry, everything else, but not biology. But I knew that biology will be at least something who will, I'm so excited about it. So I will wake up every single day to figure out what I'm doing related to biology. And of course, with all of the technology we have now, please use it. Use it to figure out what are your dreams, to figure out how you can achieve them, and to figure out how you can build actually those dreams as well. So now I will talk about what I did during my PhD in immunology. So as I told you, the PhD that I did was in the research laboratory. And in general, we have a context, which is really important. It depends where you do, on what you will work on. And you need a pretty strong background or context, how we say, because it's really important. Based on this context, it's also the next step will be to have a critical question that will help the field you are working on to move forward and figure out different objectives. With the question you have to define also different approach, method, or technique that will help achieve your potential question, because of course you have a big question, but in this question, this critical question, you have many others. And it's important to define the different approach, the method and the technique you need for that. And based on the technique you use, the method you will do, you will get some results, which could be in the way you wanted, because of course you need to have a certain hypothesis because you had a context and a critical question. Now you should look at your results and check them, see if they go along with your idea or if it's totally the opposite. of course, you have to analyze, and that's the way you analyze. Actually, we do a lot of analysis are in the computer, but I didn't say that at the beginning, but the technique we are using are mostly not on the computer, but the analysis could be. And of course, after that, I think Dr. Kayla and also Dr. Alima say that yesterday and today, you have to publish after, because it is a way to share research. You publish a paper which will help others to figure out the context as well. And of course, as you know, it's really important to understand those papers. I know that with the technology now, a lot of people share information without a very strong context on internet, but you have to be careful when we talk about science. It should be reproducible. It should be also shared by different teams to really know if your results and what you are showing are really important or are accurate, actually. So it's what I did during my my PhD. And to be more precise, I work on immunology. And I think yesterday you had an introduction about immunology, which is the study of the complex system of cells, tissue, and organs, which is the immune system. And this immune system, it's able to recognize material which are foreign and to neutralize, eliminate, or metabolize them. This it's called immune response. And in general, it's without hurting the host tissue. The host is us, our body. And you know this picture. And in there is many actors in the immune system. And in general, those could be metabolites, but the main actor are in general cells. And those cells have different abilities, and they are grouped differently also. And the main cells that I'm looking at are in the adaptive immune system. I'm just curious, who already heard about B cells or T cells? You can put on the chat if you heard about that. Okay, I'm seeing Danielle. I'm sure he's not the only one. So yes, those cells also are called lymphocytes. And sometimes you will hear about lymphocyte B, lymphocyte T, and B and T are, those names are exactly where they're coming from. The lymphocyte B are coming from the bone marrow, and the lymphocyte T are coming from the thymus. Okay, Jenny also heard about it. Okay, and so the B cells that are some lymphocytes, they are known to produce antibodies. And I know a lot of you heard about antibody, but let me know, because just to know what is your background. Did you already hear about antibodies? Okay. Carla also, that's great. Okay, okay. So many of you already heard about antibodies. So B cells, the lymphocyte B, mainly produce antibodies. And I will talk about antibodies later. And for lymphocyte T, there is different kind of lymphocytes. We call them CD8 or CD4, which are different type of lymphocyte T. And in this immune system, those cells are specialized for different functions. And as I say, the B cell will produce antibody. The T cells will recognize protein from a pathogen, which are in general presented by dendritic cells or macrophages, which are a part of the innate immune system. And those T cells will either kill the cells infected with the pathogen, or they will secrete other molecules that will add a certain inflammation and a certain response to get rid of the pathogen. And that's exactly how the immune system, like, briefly work around the adaptive immunity. And one thing I didn't say, it's a difference between innate and adaptive. It's the innate immune system. It's in general non-specific, and its defense mechanism. It's the first barrier after an infection or a presence of a non-self element in the body. And adaptive immune system, it's acquired. And we will see that in this adaptive system, there is kind of a memory. So cells remember when and what you got in the past as infection. And do you know what is happening if the immune system attack our own tissue?
Sometimes it accidentally attacks our own tissue while trying to fight off a virus, but eventually our body should repair itself.
Yes, sometimes, but when it's not repairing what is happening. Oh yeah, sorry.
Comment in the chat saying, I think one effect could be cancer.
Yes, sometimes when the immune system it's not well regulated, it could lead to cancer. But the main point that I wanted to point out, it's when the immune system attacks our tissue, it's called autoimmune disorder or autoimmune reaction. And it's one part of, it was one part of my PhD in immunology. So I work on multiple sclerosis. Oh yeah, exactly, Skylar, autoimmune disease. So maybe some people heard about multiple sclerosis. Do you know what is it? Someone can tell us before I'm saying.
Actually, I think we talked about it with Dr. Stephanie. I think multiple sclerosis is a multifactorial gene, I assume. And I know sclerosis is like a disease that comes in your back, but I'm not too informed, but that's what I remember from Dr. Stephanie's teaching.
Yes, that's one part of that. It's a multifactorial because there is some gene implicated in the multiple sclerosis. It's an autoimmune disease. So that means there is a component of the immune system that are inducing the disease. We don't know if it's the main point. We don't know if multiple sclerosis is a result of the immune system and balance, but we know that some part of the immune system attacked actually myelin, which is a protein, if you see, surrounding the nerve. And in people having multiple sclerosis, there is this shield, which is the myelin, which will be damaged. And for people having multiple sclerosis, it's really difficult to walk. It's difficult to think sometimes. They have a lot of migraine, and the motor movement are also impaired. And why? Because of the damage in the myelin, which is really important for all of the reflex movement and other things, such as the digestive system, how it's working. It's also related to the brain. So multiple sclerosis will be a damage in the brain. And this, during my PhD, I was working on different hypotheses to understand what is the immune component of which immune component is actually actor or critical to induce multiple sclerosis. And here I just put different type of like the different hypotheses that could be in that could lead to multiple sclerosis. And one thing also that you should know is in research, everything is not just black and white. It's important to look at your hypothesis and answer your question. But of course, it depends on the method you use and the approach you have. So that means you can try to look at the same question with different approach and get different results. That's why the scientific community and the research, you need to publish, to share, and so we can talk about the different aspects of the same question. That's why there is different hypotheses about multiple sclerosis, and I was working to some on that. For example, the thing that B cells can recognize the myelin, because B cells also, the lymphocyte B, can recognize a protein, and communicate with the T cells, which will attack this myelin and damage it. And another hypothesis is also because I told you immune cells can release different chemicals or molecules, and they will release the molecules which will induce an inflammation and destroy the myelin. Of course, B cell also, which produces antibody. I will tell you later, antibody has the capacity to recognize an antigen, so a part of a pathogen, and neutralize it or destroy it with a different type of mechanism. And it's one hypothesis also, and there is many others. And of course, there is no treatment, because when you get multiple sclerosis, it's something that you will have all of your life. And it was really interesting what I did during my PhD during 3 years. I also work on multiple sclerosis during my master, and it was really interesting, but I know that something was missing during my education. Oh, I'm seeing Kayla, sorry. Dr. Kayla has a question, maybe?
I didn't realize I was still up. I was just pointing out that Daniel summarized what is, it damages the nerves in the spine and brain, so that the messages aren't sent well through those nerves.
Exactly, Daniel, perfect. So something was really missing, because I know that the study of multiple sclerosis was really important because it's touching a lot of people in France, but I wanted to work in something bigger than multiple sclerosis and autoimmune disease. And I have a question actually for you to move on. So what is the deadliest animal on Earth? Do you know? I put some some options, and you can just write on the chat if you have any idea about which one will be, like, is it a lion? When I say deadliest, it's the one killing the highest number of people per year. We have a lot of G, E. G. Okay, none of them. Okay. We have winners. So before I'm answering the question, I have another question. So I put on my slide malaria, dengue, West Nile, yellow fever, Zika, Japanese encephalitis. Can you tell me what is the common points between all of the world you see in front of you?
This is a theory. Don't they all come from mosquitoes? All of
Yes.
these come from mosquitoes.
Yes. Okay. It's super easy. Oh, I like this group. You are. Yes, and a lot of others also. Leandra, Jad, yes, Stephanie, Ariel. Yes, a lot of others also talk about mosquito. And exactly, it's a mosquito. So all of those diseases that I put on the slide are mosquito-borne diseases. And actually, to answer the first question I asked, mosquito are the first animal on earth killing the most people. And according to the WHO, every year there is approximately 1 million person dying from mostly malaria, but also other disease such as West Nile, dengue fever, and you will see later Japanese encephalitis, yellow fever, still, even if there is a vaccine. So yes, the mosquito are super small, but we should be scared about them more than being scared for lions. So now I want to talk to you about flaviviruses. You don't have to, maybe it will start to be a lot of information, but just please ask me questions if you don't understand. So flaviviruses is just a group of viruses that are almost similar. And in the previous slide, okay, Daniel.
Quick question. How do the mosquitoes carry so many viruses? I know like they'll be immune to some of them, but it doesn't make sense to me how they carry so many viruses without being affected by one.
I think it's because of the cells they have, which are really susceptible to a lot of viruses. And they have their own type of virus that could kill them, but actually the one that they are transmitting to humans in general are not killing them.
Okay, thank you.
Actually, I have a very quick question. I'm so sorry. Doesn't that mean like if there's like a big, let's say a big change in the ecosystem, right? Then wouldn't we, and then it causes that there's an overpopulation of mosquitoes and insects. And since we have so many, wouldn't that cause like mosquitoes to take over half of the population of people? Because it would be like, and we would almost all die of mosquitoes if they carried that much diseases, right?
Because they carry the disease, but as you will see with the slide that I will show later, we can be infected with those virus, but we will not die because of our immune system. So there is people dying, of course, but it's we have vaccine for a lot of different disease. And of course, the mosquito continue to reproduce, but there is also a lot of techniques and methods to avoid being I don't have the word, bitten, no.
Overpopulated, maybe?
Sorry?
I'm just thinking because like overpopulated would match that sentence, like being overpopulated by mosquitoes.
What I wanted to say, it's like the transmission. The transmission also every time is not effective. So you can have a lot of mosquito, but it's not every time that you have a mosquito bite that you will be infected. And there is kind of the immune response, which is also good for a lot of people. You will see in my slide later. So I know that with the climate change, of course, there is more mosquito in some area that there were not before, but I don't think we can have overpopulation of mosquito. There is kind of, there is a balance, of course, and it's also in a lot of country, one thing that I didn't say, it's seasonal. Mosquito are not living every time, and they are not carrying the virus every time. So there is this kind of high transmission rate during raining season in some part of the world, and it won't happen during the dry season, for example. So it's depending.
Yeah, and I think for a disease like malaria, the transmission is only done from the female, right?
Yes.
So not all mosquito will carry, like the male can have carry the infected cells, but will not transmit it. It's only the female that will that is able to transmit those. I don't know about the other disease, but yeah.
That's
Yes,
because
for malaria is that, yeah.
that's because only females take human blood, so then they can feed their eggs.
But don't they die right after birth?
Oh, for
the female?
The female one, I don't know for the.
I don't know. I haven't heard that.
Yes, that's a good point.
That
I
would
can
be a
check
cool
that.
fact to know.
Yes. And just to talk more about the flaviviruses, such as it's the one, except malaria in my previous slide, dengue, West Nile, Zika, Japanese encephalitis are all flaviviruses. They are from the same family and have approximately the same structure. So it's the RNA genome coding in general for a big polyprotein, which could be cleaved in different parts. So we will have structural protein, and you can see on the, if you see my arrow here, these are the structural proteins. So on the surface in general, you will have the E protein, which is recognized by the antibody as it's on the surface. And inside you will have non-structural protein, which they have a different role in the machinery of the virus. And all flaviviruses are similar structure. And when we talk about epidemiology of dengue and Zika, because my work actually was focused on dengue and Zika. When you talk about dengue and Zika, you can see on the map, this map, it's not 100% accurate because there is some country that have outbreak of dengue, but not reported on time. And also, if you look at dengue and Zika map, you see that there is an overlapping in some continents. So some country will have both of the viruses co-circulating. And I will talk more about Zika later, because I think it's kind of fascinating to see how different Zika is compared to dengue. But when we talk about dengue, every year there is half of the population is at risk of infection by dengue. And we also wrote that there is 4 different serotypes of dengue, which is also kind of tricky for research, because one, it's already a lot of work, but 4 different serotypes, it's a bit more complicated. And among those people at risk of infection, there is every year 390 million of people who get infected, but a lot of those people are asymptomatic. So you will not have any fever, any rash, and but you will still be infected. So your immune system will react a certain way, but at a certain threshold that you don't show any symptom. And among those 390 million of people infected, we have a portion that will show symptoms, and those symptoms are called dengue fever. And do you know also why long time before it was called breakbone fever? If someone already has malaria, you may know why it's breakbone fever.
Does it weaken your bones or make you feel like tired so you can't walk?
Exactly. It's weakened. Yes, Leandra, you say the same thing. Your bones you will be very tired because you have pain in your muscle and your joint also. So it's almost impossible to stand. And that's why they were calling people, because people were not able to really walk properly. So they were calling people before having dengue breakbone fever. And among the people showing symptoms, there is a portion of people that will also develop severe cases. And in general, it's hemorrhagic fever, so a failure in some organs, and 40,000 of people will die from dengue every year. And about Zika, I think a lot of you heard about it. Mostly Zika, I will talk about that a bit later, but the thing that we remember from the TV, for example, is the microcephaly in some infants born from mother infected with Zika virus, or Guillain-Barré syndrome. And I think maybe you don't know about it, but it's kind of similar to multiple sclerosis. It will be, the myelin will be damaged and it will induce a paralysis, but it has been shown that there is an association between Zika infection and Guillain-Barré syndrome. And of course, so far, there is no efficient vaccine and antiviral drug against dengue or Zika. More about Zika, this is just for you to bring something at home and know and tell to your parents or friends that you learn about Zika, and also to show that maybe you heard about Zika in 2016, but Zika has been isolated in 1947 in Uganda. It's not a new virus, but it's emerging. So that means it was already known, and at one point something happened and an outbreak was there, and it's what happened in South America. And of course, the name Zika is also, just for your information and curiosity, is coming from the forest where the rhesus monkey was captured in Uganda. So what happened between 1947 and 2016? We didn't hear a lot about Zika because it was a lot of small outbreak. And also it was in some country where the research was not, and the epidemiology was not, I will not say good enough, but at least internationally reported. So that's why we didn't hear a lot about Zika outbreak. But of course, in the research group, if you work on viruses, you know that Zika is not a new virus, and some outbreak were already reported in 2007, 13, and after in 16 in the Americas. So as I say, Zika is a vector-borne disease with a particular mosquito, because there is also different species of mosquito, and of course they don't carry the same virus. It's really complicated, because, and in general, it's the one a bit tigered that we carry Zika and dengue. And even among the family of flaviviruses, there is some particular mosquito that will carry other virus from the flavivirus family, different than Zika and dengue. So it's really different and more complex. And because of research, and this is the importance of research actually here, when Zika, the outbreak was so big in South America, so a lot of researchers came together, and they figured out that it's not only a vector-borne disease, so the transmission could be also sexual, because they show that the presence of this virus in different fluids, body fluid, and the transmission could be also vertical, so from a mother infected to a fetuses. And all of that was because of the common efforts of researchers to understand the different transmission. And that's why it was not a pandemic, but it was an outbreak that touched different countries, not just Brazil, but a lot of other countries in South America, because of the different transmission. And it's important when you study a disease to know what are the different routes of transmission. It will help for public health measures. And it's important also to know that as dengue, a lot of people are asymptomatic to Zika. So that means someone asymptomatic can, because of the other routes of transmission, has 80% of people are asymptomatic. The transmission can continue, but in silence. So that's why it was kind of tricky during the outbreak also. And of course, has the 20% that will show symptom. I just put the common symptom, and you will see that among a lot of viruses, the symptoms are pretty common: headaches, rash, muscle pain, joint pain, or fever, red eyes also. Any question? Okay. I hope it's interesting. I'm sure it's interesting.
It's really interesting.
Oh, thank you.
Very much so.
And just to know more about the transmission between mother and fetuses, how the figure out that it was one of the route of transmission, it's because when they look at epidemiologic data, and this is a lot of computer work, because the group all of the data from patient, and someone it's making graphs and looking at association. And it's something also that could be done by a bioinformatician. And they look at the population infected, and they figure out that there is an increased incidence of fetal abnormalities in fetuses born to mother infected with Zika. And at the end, it was called congenital Zika syndrome. And it's not just microcephaly, but I will also show you what could happen. And the research also showed that when Zika happened, when the infection to the mother happened during the first trimester, is increasing the risk of fetal abnormalities, because Zika has a tropism. So when we say cell tropism, it's when a virus like a certain type of cells. And Zika really like cells in the placenta. We don't know why, but cells in the placenta are very susceptible to Zika. And in general, it's also possible that during the first trimester of pregnancy, the barrier, because there is a barrier between the mother and the infant, and the fetus, but this barrier is not strong enough during the first trimester. So it's possible to exchange things like viruses. And when we talk about microcephaly, just to show you that, as I say, is important when you talk about, when you want to show association, is important to have it in different parts of in different areas, but also published by different persons to really be sure that your idea of what you are seeing, it's accurate. I think Daniel has a question.
No, I read it properly and I saw something. It's okay now.
Okay, thank you. So in Brazil, during the outbreak, they show a lot of cases of microcephaly among infected mothers. And the same thing was shown in French Polynesia by retrospective study, because as I show you in French Polynesia, the outbreak was in 2013, but the just stock data, they didn't analyze it really. And when the outbreak in 2016 happened, they analyzed it and they figured out, yes, microcephaly was actually associated to Zika infection. And even in US, it was happening, because I think in Florida, there is some mosquito that can transmit Zika in Florida, and it has been reported also there. And as I was telling you, microcephaly is not the only problem that could happen when a mother gets infected during pregnancy. There is other symptoms such as hearing loss, visual deficit, seizure, or learning disability. Because when I was talking about the tropism of Zika for placental cells, it's also the case for brain cells. Zika has like also to infect the brain cells. That's why it's doing all of these outcomes. And the other problem linked to Zika, I talked to you about that, is Guillain-Barré syndrome, which is a severe acute paralytic neuropathy with progressive muscle weakness. And as you see, it's also a damage of the myelin and has there is different theories about Guillain-Barré syndrome. People think it could be autoimmune, but also it could be linked to a virus, a viral infection. And among the different virus that has been shown to induce or at least trigger the Guillain-Barré syndrome, ZIKA, it's one of them. And there is different symptom linked to the damage of the myelin. And unfortunately, 5% of the patient will die from respiratory complications. So that's pretty sad, but there is a lot of research going on to figure out how to prevent or how to understand Guillain-Barré syndrome and avoid that after Zika infection. Yes, Danielle, you can talk.
If you are a smoker, would that make you more susceptible to death from the Zika virus?
If you are a smoker, is
Would
what
it
you
make
say?
you, yeah, would it make you more susceptible to death from the virus?
No, there is no study so far. There is no study about smoke and Zika infection, and because Zika is not attacking lungs, so I think that's why the researchers are not focusing on looking at smoke, any association between smoker and Zika infection. But of course, smoking will damage more than just your lung in general. And there is, for other disease, there is a pretty bad association, negative association between smoking and those diseases. So if I can give an advice, it's don't smoke.
I wanted to ask, so my understanding was that smoking can affect your immune system. So would that potentially make it easier for just any disease, not specifically Zika virus, because you said they haven't tested it. So like until we test it, we don't know, but we could hypothesize or speculate that it could affect death rates.
Yes, I agree with you, because it's of course, smoking will modify your immune response. So if we extrapolate, it could modify your response against any viral infection and make you more susceptible. So right. Yes.
But like you said, we don't know that for certain.
Yes. So another question for you. Do you know how to study actually the immune response against viruses? There is different way to study the immune response, but someone can tell me what are the different way we can use?
Is one symptoms?
Sorry, say that again.
Is one of the way to study viruses symptoms?
Yes, we can look at symptoms. And if we talk about like methods, how can we look at the immune response in human, for example? Anyone?
You used the word antibody before. Is that relevant?
Yes, but where do we find those antibodies?
B cells.
And those B cells? like what is the component of the body we can use to look at these immune cells when we want to study it?
With the
Is it
blood.
something that we can easily get?
Yes.
I see Leandra says white blood cells.
Yes, and we get them from blood. I know my question was a bit too vague, but yeah, it was just to say that to study the immune response, because human, we can give blood, but in general, the immune response is not happening only in the blood. It's happening in different organs, and it's really important to look at those organs, but we cannot because, for example, our spleen, it will be difficult to look at the cells in our spleen, but spleen, it's really one of the main organs of the immune response. So that's why it's important sometimes to use different models to look at the immune response against viruses. And I'm not working on that, but I know that it's possible to use non-human primates to do research. And there is, of course, pro and cons. But all of one thing I really want to highlight, and one thing that you should know, is every research working on animals, it's very well followed and regulated. So you cannot do whatever you want with the animals, and all of the ethical considerations are taken, because animals are not suffering, or very less, and everything should be justified also when we use animals. And so it's possible to use non-human primates, and this will help to evaluate vaccine drugs, viral tropism, and more. It's also possible, and this is where I am and what I'm doing, it's you can work with different mouse models. So mice are also, they have pro and cons, but in general, it's one of the most used animal model to study viruses. And it can be used for vaccine, drugs, viral tropism, pathogenesis, sexual transmission, or congenital infection. And one thing actually that when people were using mice, for example, we figured out that ZIKA and also dengue are viruses that modify actually our immune system when they want to infect our cells. They are pretty smart because they know exactly what to shut down to be able to infect our cells. And that was known because it was impossible at the beginning to infect mice. And we figured out that it's because ZIKA recognize one of the proteins from the ZIKA or dengue, the NS5, will recognize a specific component of the immune response, which is really key for inducing an immune response against ZIKA. ZIKA will shut down this component and will be able to infect and do whatever he wants in the body. So it's it was pretty nice to figure out that and be able to modify mice now to use them and learn more about ZIKA. Any questions? And Dr. Stephanie, I'm just wondering how long I have, I still have more slides, but I just want to see if I should go faster or just points certain things.
Yeah, so far we have about 15 minutes till the end of the camp, so we can just continue with if there are questions, they can start asking. Otherwise, this time is the time for discussion, is a discussion session. So
Yes.
if you guys have any question, that would be the time. Other than that, I don't know. You can go ahead. Does anybody has any question?
Okay, I will just continue and feel free to ask questions. We don't have to do all of the slide. It's better if you have questions. I will answer, no problem. Don't worry if I'm still talking. You just have to unmute yourself and talk.
Okay.
Okay, and here I put one example how to use mice actually to answer, for example, the efficacy of vaccine candidates. So in general, you will use when I say prime or boost, I think a lot of you maybe heard about it with the vaccination going on for the pandemic. Prime is the first time we give the vaccine, and a boost is when you get a second shot of the same mixture of your vaccine. And we do the same in mice. And of course, we use blood and different organs in mice. But one thing that we use to see an efficacy, so to know if a vaccine will be good or not, because of course you have different candidates of vaccine and you have to figure out which one you should focus on. We look at weight loss of the mice after vaccination plus infection with the pathogen we want to prevent or cure. And when you look here, you see saline in general, it's the mice that were just injected with a solution, a saline solution that used like control. And we compare that with other mice injected with our vaccine. So after infection, in general, the infection is at day 0. Every day, so daily, you will look at the weight of the mice and compare with the starting weight and see if there is a decrease, because the loss of weight, it's one aspect of the disease. It's showing that the animal is starting to be sick. And we also have, you don't have to look very precisely to those clinical score, but it's also another way to look at the disease in animals. With clinical score, we will see, for example, I told you that a human, when you are symptomatic, you show different type of symptom. It's the same. With mice, it will be the weight loss, but also they will start showing different things. For example, the fur will be not really smooth. They will start to move, to have less mobility. And based on that, we give a certain clinical score and we will know if the mice are getting sick or actually if the vaccine prevents again sickness. And that's one of the way a lot of people, and actually it's the first step to look at efficacy of a vaccine candidate. And of course, after you can do different type of animal model, and after you go to human, and we call that clinical trial. So currently, my area of research, just to show you what I'm doing, it's I'm trying to define the role, so the importance of the T cells, which is, I talked to you about that, it's some immune cells. I want to know what is their importance during viral infection. It's those cells that we call T cells are the main one that fighting against viral infection or not. And one other aspect of my research is also to look at the different components of the virus that will trigger the immune response. And I show you that at the beginning, the big polyprotein that it's encoded by the genome, it's of ZIKA. There is structural and non-structural, and it's important to look at which protein of the virus induce actually and trigger a big immune response, because this way we will understand which part of the virus we have to give to people to induce the immune response and prevent infection. And this is a concept of vaccination. And of course, all of that will help me to develop and test innovative vaccine approach against emerging viruses. And I also working on with human samples and people in endemic countries, because of course we are not endemic for dengue in US or Zika. I'm working also on other diseases which are not endemic in the US yet, because we never know when it can come. And I'm working with people in the endemic countries, because some endemic countries, they have very good researchers, but not enough cutting edge technologies to be able to really look at the immune response and to understand the virus around them. And I'm initiating collaboration with people in those countries. We identify immune signature, and this is where bioinformatics is coming, because it's a lot of technique called RNA sequencing, and it's at the level of a single cells that we look at the up or down regulation of different genes, like a bunch of genes. And this way we can understand if people infected with a certain virus can have a particular signature. And based on that, we will figure out which vaccine or which therapy it's good for them. And of course, it's important because I don't know if I told you, but the immune system interacts a lot between, there is a lot of interaction between cells. And it's important to look at the interplay between antibody and T cells for my type of project, but others also are working on other interactions. And all of that, it's really to investigate the precise role of adaptive immunity in protection versus pathogenesis, because the immune system, it's really well built, but of course, as everything in life, there is a balance that you need. And when there is disease, that means something is unbalanced. And at one point, the same cells that protect you could be a source of, could be a danger for you. And it's, in viral infection, it's important to know what is this balance and when a cells can be protective or pathogenic. And it's exactly what all of my projects are about. It's the main goal. And I think I will go faster for that, because here I was just showing that antibody, if you look at the right part of the slide, neutralization assay, it's one of the techniques we use a lot to see if an antibody is good or not. So from the blood of patient, we are able to look at the neutralization capacity of an antibody, because one of the function of an antibody is neutralize a virus. And in vitro, meaning that when we take cells outside of the body and we work on that in plate or flask, if we prevent infection, as you can see here, if we prevent infection, that means the antibody are good and are able to neutralize the virus in vitro, or if they don't recognize this virus and don't neutralize, we will see an infection. And that way, with antibody, we are able to look at your history. So I can take the blood of someone, in general, the serum, so the part, the clear part that we obtain, which is full of protein after a centrifugation of the blood. In the serum, if I look at some of your antibody, I will be able to let you know if you had a previous infection against one or another viruses. So it's pretty good technology. Antibody can be a marker of previous infection. And here I was just showing that all of my research, it's really important to design safe and efficient vaccine. And I hope that it will be applicable to several other infectious diseases that are threatening in the world. And one thing also that I'm doing now during the pandemic is to look at, I told you that flaviviruses, for example, it's a lot of different viruses that are kind of similar. They share some of the protein, have a homology. So the sequence, it's almost similar, sometimes 50%, meaning that your immune system can recognize another virus because of this cross-reactivity. It's the way we call that. So it's important to know how does prior exposure to a virus can influence the outcome of a subsequent infection. And I think this is a beautiful virus, but I think everyone saw that beautiful. I mean, the microscopy, this they did a good job, and I think everyone know about it. Can you tell me?
It's COVID-19.
SARS coronavirus 2.
Exactly. So COVID-19 is a disease caused by SARS-CoV-2. Actually, this virus is called SARS-CoV-2. SARS-CoV-2 means severe acute respiratory syndrome coronaviruses. And why 2? It's because in 2013, the first one actually had also in the Middle East made a very huge outbreak, but it was pretty severe because the ratio of dead people was way more higher than what is happening now. There is not a lot of survivor for SARS-CoV, but it's not the case for SARS-CoV-2. It's less at least, and because of the similarities on the sequence, I'm talking again about the sequence, they call it SARS-CoV-2. And when I was talking about previous infection, SARS-CoV-2 is from the family of coronaviruses, and other coronaviruses that we have every year here actually are the human coronaviruses, which cause common cold. Every year we have running nose and like sore throat, and it's one of some of these, it's caused by some of the human coronaviruses. There are seasonal, and we know already 4 of them. And it's important to know if a previous infection to one of those human coronaviruses is beneficial before SARS-CoV-2, or actually it's harmful for harmful meaning if you have a previous infection, common cold, maybe you will worsen your subsequent infection with SARS-CoV-2, but it's still ongoing. There is not result about it yet. And yes, I just wanted to let you know that it's possible to do internship during the summer. We already in my lab have high school students. So, and it's really important, take this opportunity. We have multiple laboratories in La Jolla Institute. I'm a part of infectious disease and vaccine. So let me know, or just send an email to Dr. Stephanie and she will put you in relation with me or La Jolla Institute directly for any internship, because it's really important to do that during high school. It will help you.
Denny is asking, do you guys accept middle schoolers?
Middle schoolers. I have to check that. I don't know if there is an age limitation. I have to check that. I will let you know.
I also would like to ask, do you accept non-US students?
Of course, yes. I have, it was not high school so far, but I had a lot of people coming from Asia, Europe. I don't have Africa yet, but coming to do a master internship or just to exchange of technology and to learn about one technology in immunology. Yes, it's possible. And I really like when Dr. Stephanie say yesterday what she said about doing remotely an internship. I think it's really important that we open this kind of door, but the only problem, as you saw in my presentation, is a lot of lab technique. But I'm sure if we look deeper, we can find it's possible to do an internship remotely, because I know that it's not easy to come to San Diego for some. So yeah, I will think about that, and I'm sure I can make a plan for that. And yes, here I was just putting all of the disease we have in La Jolla Institute, at least the one we are studying. And yes, thank you for your attention. Thank you for the discussion. Thank you for listening. I'm really happy for this boot, doing this bootcamp, and I hope we can do that every year.
Okay,
and next
so
time I will
everybody,
show you.
If there is no other, oh, and we had a cat, yes. So everybody join me to say thank you to Dr. Annie and Dr. Kayla for talking to us today. And we're very, very happy you took the time to do this. And I hope that all of you learned something today. So big shout out to our presenters today.
Thank you, everyone.
And this is the end of day 2. We hope to see you tomorrow. And let us know if you have any question. If you have any question, drop them in on Slack. There are some activities there. You can spend some time playing around with the data. Dr. Kayla put some very interesting video there. Take a time to look at it. And yeah, we're here. We're going to be answering your question if you have some. And thank you for your attention.
Sorry, I have a question for Dr. Annie. So
Yes.
I'm not sure if it's the same in the USA, but in England you take the flu jab every year. Why is that if our body creates antibodies?
Oh, because for the flu, there is a lot of variants, and I didn't talk about it, but it's almost the same for SARS-CoV-2 now. There is a lot of different variants. So sometimes the antibody, because the variant means that the sequence is similar, but kind of different at a certain point. So the antibody for one strain will not be stronger enough to neutralize the next strain. And for influenza, there is a lot of strain coming almost every year that are different. That's why we need the flu shot almost every year.
Merci. Au revoir.
Au revoir.
Au revoir.